In this video, Steve Boswell, ISPE UK Central Region Member and Director of SV Process, talks to Trevor Page, Group Development Director at GEA, about the future of pharma continuous QBD manufacturing OSD.
So what do you believe are the challenges facing the industry in adopting continuous manufacturing?
For the reasons of cost, quality, compactness of process, etc. Is it a paradigm shift in pharma? Yes, I think it is to an extent. Not because the process is that revolutionary, but because the thinking behind it is a big shift, particularly how you test the product and how you assure product quality and how you develop the process.
So is that a regulatory issue as well?
The regulators themselves seem remarkably relaxed and have recently said quite clearly there's no need for a change in regulation. What seems to often be the difficulty is the way regulation is interpreted. You need people who think in a slightly different way.
And now let's talk about continuous manufacturing, the advantages of it for manufacturing and for process development.
Well, I guess the way I see it is the regulators simply want to make sure that every patient gets good quality product with the right dose in it. And if you look at the way we have traditionally made materials or made products in a batch process, we assume that the whole batch is homogeneous in the vessel, and we take a few samples. What I think becomes clear in a continuous process is, firstly, you have the ability to adapt the process while it's running to respond to external influences, external environmental changes, or even raw material changes. And the amount of data you get per kilogram of product means that you're actually analysing, or potentially analysing, a lot more product and building a lot more robust understanding of the process, where you can build distributions which start to tell you with great confidence not only was what you measured correct, but actually what you didn't measure is highly likely to fall within the acceptable limits that you're looking for. The adoption of process capabilities, that's been used very powerfully to show product quality and to eliminate the need for end product testing in the polymer industry. So I have every expectation that that's where we will end up in the pharmaceutical industry, eventually.
And what part does QBD play in that, and how does it relate to it?
Well, I mean, if you look at what Quality by Design says, it says you need to understand your process. And to understand your process, you need to have somehow built a map of that process to show what conditions give you quality product. Now, if you're going to have a space in which you know the product will be of good quality, you need to have gathered sufficient data. So it's not just data at one point, it's data over a space. And that means doing more experiments, and in the batch world, that means doing a lot more individual batch runs. What we see in continuous is the number of data points we can explore in a very small space of time. So, literally within 15 minutes, we can have done almost 15 different points of granulation, looking at moisture levels or work levels within the granulation, and that will have taken us all over a range of particle size and moisture contents. And that starts to build the ability, or give you the ability to explore many more process conditions in a lot more depth, with less material and in much less time. And that means that your file of data is much richer and much more convincing.
With all this in mind, how does this relate to manufacturing technologies themselves?
Well, I guess if you look back at the history of continuous processing, I think you'll see that the sort of early discussions around continuous were often dogged by discussions of large-scale products, large equipment, etc. There was also some early attempts to produce some quite novel technology, for example, continuous microwave drying of granules. But I think that was possibly a step too far. So certainly from my perspective, it's always been very important to develop equipment that was digestible, and to basically bring it down to something that matched the production scale that was needed for pharma. And from the discussions we had with most of the customers who were interested, the sweet spot was actually quite small. It was in the 10 to perhaps 50 kilo an hour range. And on top of that, the biggest driver was to make sure that equipment was capable of getting representative material with very small quantities. If I look back at some of our early attempts in continuous, perhaps in using fluid bed dryers from the milk industry, these had residence volumes of perhaps 40, 50, 60 kilos and took a lot of material before they reached an equilibrium. What we needed to do was scale the whole thing down so that we could reach equilibriums very quickly. And this was always very deep in our mind, that we wanted the equipment that was used for development to be used directly for manufacturing. No scale up, no change, and in fact, although it's taken a few years more than we might have expected, that is now happening. And it's amazing to see a processing line put into something like an overgrown container to be used for development on one site, and then with the prospect of the whole thing being picked up and moved to a production site without huge amounts of re-engineering, with the equipment manufactured largely in a factory rather than on site, with goals of better quality, faster startups, less site work and cost.
What do you make of continuous manufacturing and the advantages to continuous quality control attributes and design space?
What I saw coming out of the petrochem industry was that we directly measured many of the process parameters, but also the product quality attributes. So typically, this was gas chromatographs used in quite hostile environments on the plant to get real-time measurements, which were then used to control the process and the product quality. And the typical problems that were often overlooked were how to get a representative sample, how you present a sample in a consistent way, the effect of sample presentation, how to make the instrument robust physically, how to make sure that the observation windows were kept clear, and this was something we spent a lot of time thinking about, how to calibrate it routinely. All of these things you see when you see them on plants in the petrochem or other industries, and we needed to map that thinking into a pharma environment.
What influence do you see with continuous manufacturing on the big pharma, ethical manufacturers, generic companies and CDMOs?
When you look at the business case behind continuous processing, to date it's been driven by saving money by speeding up development and time to market for high value drugs. And I think it's one of the things that surprised us is how much money it's possible to save when you develop a process on a continuous line versus having to develop it on a batch line and scale it up. So I think that is driven for high value products. And of course that drives the need to have this small compact equipment with low minimum processable quantities. Once you flip over to a drug that is out of patent and therefore the API costs are much lower, the driving forces change totally. And at that point you're really talking about not worrying about burning so much drug during development of a process, but actually the true efficiency at high production rates or large scale production. So I think that you can see how that drives you towards potentially different designs of equipment, different driving forces between a high value NCE and a low cost API, and hence a lower priced product where you want to manufacture in higher volume. I guess the question is, will all drugs go from low volume to high volume? Possibly not. Depends on what the therapy is for. So I think during the development process, one of the things that starts to become very clear with developing a process for a continuous process is that at the early stages you're going to have to handle the very material, sometimes the difficult to handle API, that you're granulating to get rid of. And that means that your capability to handle poor flowing materials, cohesive materials, has got to be suitable. It may well be that your cohesive and poor flowing material doesn't flow brilliantly out of a feeder. And if you look at the variability in time, it might be quite significant relative to a unit dose. But once you put that through blender, that blender acts as a low-pass filter, and the residence time within it means that the variability that you saw in time on the inlet disappears on the outlet. And we've seen processes where, for example, you could stop the API feeder for a few seconds, restart it again, and because of the back mixing, you got perfectly good assay and homogeneity at the end of that blender. Now, this comes down to a real balance between plug flow and back mixing. But if something is totally plug flow, it means that you don't have any ability to absorb variation in flow rates or behaviors during that unit operation. So understanding that relationship and mapping the process in terms of plug flow elements and back mix elements is very important. More plug flow, better for traceability, easier to remove material if you have to. And I think, at least as a paradigm, we should not be looking at systems which reject material as a method of process control. That's entirely wrong in my view. We should use rejection mechanisms only for known causes, startup and shutdown, etc. But in principle, plug flow is useful for traceability of raw materials, identities, etc. But a bit of back mixing is pretty handy to deal with the inevitable noise in a process.
What excites you about the future of oral solid dose and continuous manufacturing, and how do you view the future?
Well, what really excites me is I think we're moving almost through the first generation of continuous processing. I mean, I actually have a four-stage map that I look at, and the first stage of the map is to take processes that almost replicate what were done in batch and make them continuous. It's not the end game, but it's a starting point to get into using continuous processing, and it enables us to start manufacturing products with confidence that we have the raw materials, we have the transformational steps, and we can generate good product from that. For me, the next step is trying to eliminate what I see as the rework elements of that. So classically, wet granulation is a rework step because of the nature of the materials upstream. I see that some of the projects we're working with customers to look at how to expand the range of continuous direct compression by eliminating some of the risks of direct compression. Instead of putting a blend that is maybe mesostable into a bin and risking it segregating, if we blend it and use it instantly, we widen the range of products that can be directly compressed, and that makes the process smaller and more compact. So for me, it's all about driving the process to be smaller, more efficient, and still able to handle a reasonable range of material properties from the upstream processes. I mean, beyond that, I think that there are some real opportunities to look at the way particles are engineered, perhaps in the spray drying or in the formation, before the formation of a tablet, rather than blending, so actually drug-loaded carriers. I think there's a lot of potential there, and of course that could lead on to tablets or oral doses that are not even compressed tablets. But I think that's a long way away. I mean, I see that as relatively unfamiliar territory to most people, although we have seen the first 3D printed drug approved by the FDA. So I think what's happening is the pace of change is hotting up. People are now, the industry is much more aware that there are more ways to make a tablet than putting in a large bowl and making a granule and then drying it.